Welcome to another video to help students of anatomy and physiology classes better understand the feedback loops associated with the hormones insulin and glucagon. So the focus of this video will be to look at the hormones secreted from the pancreas in the pancreas's role as an endocrine gland. Do note that the pancreas also participates in exocrine gland functioning. The two hormones that we're going to cover in this video include insulin, possibly one of the most famous hormones and disease pathologies that people commonly are aware of, and its lesser known but equally important counterpart, glucagon. So let's get started with glucagon first. Okay, so we're going to start with considering glucagon, which is the lesser known of the two hormones that are produced by the pancreas. And the hormone glucagon is largely secreted in periods of famine, when somebody is fasting. Essentially, this is the hormone that is predominantly used in between meals to help a patient, to help an organism survive in those gaps between when they're able to eat. So the clue, the cue, the signal that glucagon is responding to is when blood glucose levels begin to fall. Now, up here I've illustrated a pancreas. The pancreas has a head, which is this larger, rounder end, and a tail as well. It sits nestled underneath the stomach. The head is in the curve of the duodenum, and this is going to kind of rest in your upper right-hand quadrant. This organ does serve exocrine function jobs. It's going to assist with digestive enzymes, which is why it's located where it is located. But our focus is going to be on the endocrine portion. And to better understand the endocrine portion, we need to recognize that then the pancreas is divided into cells that are going to function in the exocrine component or function in the endocrine component. The cells that are important to us for our intents and purposes are going to be located in something that used to be called the islets of Langerhans, but now it's referenced as pancreatic islets. And in the pancreatic islets, there are a couple of different cell types, more than what is illustrated here, but we're going to focus on the alpha and the beta cells that are in these pancreatic islets. So when blood glucose levels fall, these cells get signaled. And for glucagon, we're going to focus particularly into the alpha cell. There is going to then be glucagon release from alpha cells. So that is our first hormone that we see coming out of the pancreas when blood glucose levels are low. So we're going to make note that an increase in glucagon is occurring when the alpha cells are releasing glucagon in response to low blood glucose levels. Now, glucagon and insulin are opposite hormones. So when one is busy and being released, we want the other one to be turned off. So there's a little bit of a paracrine signal that happens within this area, and the alpha cells get to tell the beta cells that it's time to decrease insulin secretion. So we see an opposite effect taking place here. The key to all endocrine systems is that we have to have not only a functional signal, but we also have to have receptors. And for glucagon, there are two tissues that have receptors. The two tissues that have receptors for glucagon are going to be the liver and fat cells. Now, I always illustrate fat cells with their nucleus shoved to the corner of the cell, to a pocket, because fat cells are going to be so full of their stored molecules in vesicles that there is very little room left for their organelles and their nucleus. The function of glucagon, when bound to receptors in the liver and the adipocytes, are as follows. First, the liver is going to take glucose that it had stored previously in the form of glycogen and break that glycogen down into glucose to be able to liberate it into the bloodstream. Fat cells that are bound by glucagon to their receptors are going to respond by increasing lipolysis and breaking down that stored fat into molecules that can be converted into glucose. The goal of this then is to increase blood glucose levels. So as the liver is breaking down glycogen into glucose, that's going to help release glucose into the bloodstream to elevate blood glucose levels, bringing us back to homeostasis. At the same time, the action of the fat cells is further supporting the development and creation of blood glucose to bring the body back to homeostasis. Key point here is that glucagon has receptors in two tissues, the liver and the fat cells. And when we look into insulin, we're going to find that there are only 3 tissues that insulin is going to bind to. So be watching for that when we move into the insulin feedback loop. It is vital that we respond to low blood glucose levels. And as such, all bodies have multiple ways to respond to low blood glucose levels. While glucagon is a key component to this, there are other hormones that play a role as well. Other hormones that are crucial to supporting blood glucose levels include epinephrine as an acute stress hormone, cortisol, which is important to elevating glucose levels prior to arousing the brain in the morning, but also as a long-term stress hormone. Other hormones like growth hormone are continued to be secreted even through adulthood in order to support blood glucose levels, in addition to those stress hormones and in addition to the glucagon. Additionally, the autonomic nervous system is also going to be monitoring situations and can respond by facilitating sympathetic division. And when the sympathetic nervous system is activated, a neuron from that sympathetic nervous system can head right out to an alpha cell and tell that alpha cell, you know what, we need some glucagon because our blood glucose levels are too low. So next, we're going to consider what insulin does in those situations where we have elevated amounts of blood glucose because we've just eaten a meal and we need to get glucose levels in the body back to a normal range. All right, so insulin, that's going to be the goal of this next illustration. So we put insulin up in the corner, and the reason that insulin is secreted is to help bring blood glucose levels back down. So this is going to be the hormone that is released just after a feast. This is our post-meal hormone to take all of that energy that we just brought into the human body and store it for all of the much more likely situations in which energy is depleted. So again, we have homeostasis up here as our net goal. We have our pancreas with our alpha and our beta cells in our pancreatic islets of Langerhans. And in this case, when homeostasis after a meal is involved, we're going to see an increase in blood glucose levels. And that is the key. Note this time I'm drawing that arrow to the beta cell, and the beta cell is going to respond with its first primary objective to release insulin. Recall that the alpha and the beta cells are producing 2 different hormones, and as such they have opposite effects. So when the beta cell is active, it's going to send a signal over to the alpha cell, and the alpha cells should reduce the release of glucagon into the system. So to summarize, when increased blood glucose levels are detected, this is going to tell the pancreatic islets to activate beta cells to release insulin. Put a beta up there as a reminder. And for the alpha cells to lay off their production of glucagon, put an alpha there to remind us that that's going to be that inhibitory. This way, these hormones maintain opposite effects. So now, where are the tissues that have receptors for insulin? Okay, so when insulin is released, 3 target tissues respond. There are receptors for insulin in the liver, there are receptors for insulin in fat cells, and there are receptors for insulin in muscle cells. Those muscle cells include skeletal muscle cells as well as heart tissue. And when the receptors are bound by hormones in each of these three tissues, when insulin binds to each of these three tissues, we see some very particular responses that help pull the blood glucose out of circulation and store it for later. Two of these tissues have the ability to store glucose in the form of glycogen. So glucose molecules are integrated in the cells to form a more complex sugar substance that is able to store that sugar until needed later. The two tissues that do this are the liver as well as muscle cells. You may be familiar with glycogen stores in the heart or glycogen stores such as carb loading before a race to store glycogen into muscle cells. Now, fat cells don't store glycogen. Their goal is to store fatty acids and triglycerides. And so when there is excess amounts of glucose, those fat cells are going to absorb the blood glucose under the signal of insulin and store them in complex fats for a later time period. Across the board, if we are storing glucose, whether it's the liver or muscle cells from the heart or skeletal muscle, that's going to draw blood glucose levels back down to normal homeostatic ranges. Additionally, pulling glucose in and converting it into fat is another way that we are going to decrease the blood glucose levels. So all three of these cells that have the receptors for insulin respond by taking the glucose out of the bloodstream and storing it in a complex, larger format internal to the cell. While the pancreas through the endocrine system can be responding to elevated blood glucose levels in the production of insulin, we do want to note that there is an autonomic nervous system division known as the parasympathetic division. And that parasympathetic division is going to be controlling and also monitoring blood glucose levels and able to signal the pancreatic islets to respond in preparation. So even prior to any food hitting the system, the parasympathetic autonomic nervous system running through the hypothalamus can be preparing to introduce and to draw glucose into cells. So to summarize, when blood glucose levels fall outside of the normal homeostatic range, beta cells in pancreatic islets, or the islets of Langerhans, are going to start to secrete increased amounts of insulin. The three tissues that respond to insulin, and this might surprise you, not every cell responds to insulin, only cells that store sugar respond to insulin. So liver cells, fat cells, and muscle cells in skeletal muscle as well as in cardiac muscle are able to draw that blood glucose to their interior, convert them into more complex sugars or fats, and store them for later. This overall has the effect of decreasing blood glucose levels to get a patient's body back into homeostatic ranges. Homeostatic ranges.